Yes.
Yes, that's quite so.
It's okay. Okay.
It's okay.
Okay.
Well done.
Well done.
Good job. Go on.
Okay.
Start.
Okay. Hello, all teachers. My name is Sajjad Ahmed, and I am from Pakistan, Khyber Pakhtunkhwa. And I have applied to the Shanxi Agricultural University for PhD in plant sciences. So here's my academic background. I have done my bachelor's in botany from University of Malakand, that is situated in Khyber Pakhtunkhwa, and I was graduated with CGPA 3.56. And after that, I was enrolled in MPhil botany. My specialty was stress physiology, and the university was the same, University of Malakand. And I was also graduated with the same GPA in MPhil as well. So now I'm really thankful to you all that giving me the opportunity to interview me and to listen my the thing that I want to share. So now let's come to the title of presentation, that is effects of plant growth promoting and halitolerant bacteria on Gossypium hirsutum, growth and salt tolerance capability. So we have done this research in our MPhil work, and the presentation should be contain the following contents, that is introduction, objective, materials and methods, results and conclusions. So let's start the introduction. We have selected the plant Gossypium hirsutum, which is a plant cotton, and it is a key global crop which is cultivated in almost over 100 countries, but the major producer are China, India, USA, Brazil, and Pakistan. And cotton productivity is severely affected by abiotic stresses. More than 40% annually yield is affected by these abiotic stresses, and especially salinity. How salinity affect the crop productivity by salinity can affect the productivity by causing the osmotic stress, ion toxicity, and nutrient imbalance. As shown in the picture I have mentioned here. And so plant growth promoting bacteria offer a sustainable and eco-friendly approach to enhance salt tolerance by following that they produce phytohormones that is necessary for plant growth and their development. So that is like indole acetic acid and cytokinase, which is important plants hormones. It also solubilizes nutrients like phosphate, and it solubilizes the phosphate for the plants, and it also generating some protective metabolites like proline and antioxidants. The study aims, we have performed this research to break that gap by identifying efficient plant growth promoting strain with potential application as a biofertilizer. Let's suppose if we use chemical fertilizer, so that should lead to pollution, and it will also, they have some adverse effect on the environment. So that's why we need some biofertilizers which don't have that much adverse effect on the environment. So that's why we select bacteria and plant growth promoting bacteria especially to minimize this problem. So our objective was isolate and characterize halitolerant plant growth promoting PGFR strains. And after that, to the second objective is to evaluate their effects on cotton growth under saline condition. And the third one was assess morphological, physiological responses of inoculated plant under salt stress. So our whole material and method, the methodology was include that was plant collection. We have collected the plant from NRC National Research Center, Islamabad, and after that we also isolate our rhizobacteria, and we have to identify the identification. And after that, the third one was characterization of that isolated plant growth promoting bacteria, identification of strain, to identify the strain of bacteria, isolated strain. We use 16 RNA, ribosomal RNA sequencing, and then inoculation of plant growth promoting rhizosphere bacteria to cotton plants, and then salt stress treatment and their effect on morphological and physiological parameters. So here is our result. When we isolate the bacteria, so we just we just find out that does this bacteria resistant or tolerance to the temperature. So when we expose this bacteria to the extreme temperature, so the both strain DS2 and RT12 shows tolerant to both extreme temperature, that is 4 degrees centigrade and 45 degrees centigrade as shown in the pictures. After that, sir, we check the salt tolerance ability of this plant to assess that does this bacteria show resistance to the salt. So after checking at RT12 showed well growth on 10% and medium growth on 15%. So however, while and 20% that didn't show any growth. After that, sir, DS2, we check DS2. So DS2 have a well growth on 10%. However, while it was not shown growth on 15 and 20%, as shown in the picture, sir. So the colonies were not formed in 15 and 20%. It means that doesn't growth and this salt concentration. So after that, we check the bacteria to characterize it. We have characterized these bacteria that does it can have the properties to promote the plant growth or not. But that we checked for phosphate solubilization. So the bacteria were RT12 was found positive for solubilization of phosphate, and they were able to solubilize the phosphate for plant. While DS2 didn't show a halozone, which the halozone shows that this bacteria have the ability to solubilize the phosphate. So here in DS2, the picture, it's clearly seen that there is no halozone was found. That means that these bacteria were negative for the phosphate solubilization. After that, we checked the bacteria for ammonia and HCN hydrogen cyanide production. So the both strain RT12 and DS2 were noted positive for ammonia production, and we know that ammonia is too much important for plant growth, because ammonia is the plants cannot take nitrogen in nitrogen molecular form, and we know that atmosphere the nitrogen is present in 78%. So that's why plant cannot take in that form. So it should be converted, and plant takes it in the form of ammonia or nitrate. So these bacteria can convert nitrogen molecule into ammonia and nitrate forms by the process of nitrogen fixation. So these bacteria were noted positive. And after that, the strain T12 and DS2 were also checked out for the production of hydrogen cyanide. And these bacteria were found to produce hydrogen cyanide. After that, we have firstly identified these bacteria, sir, and these bacteria were identified, and we isolated this bacteria, and then we identified it by using the 16S RNA, and after that sequencing by using the single sequencing method, we find out the sequence, and then we search it in the NCBI, and the phylogenetic tree shows that RT12 was as a Pseudomonas honenensis, while RT12 was identified as a Staphylococcus peptide. After that, sir, we inoculated the bacteria into the cotton plant under salt stress, and both strain DS2 and RT12 increase roots growth under salt stress. Similarly, bacterial strain DS2 and RT12 shows increase and shoots growth under salt stress. Here we can see that in the graphs. After the bacteria strains DS2 and RT12 increase leaf length under normal and stress condition. And also the both strain, bacteria strain DS2 and RT12 increase leaf number under normal and stress condition. So both strain DS2 and RT12 increase chlorophyll contents like chlorophyll A under normal and stress condition. And also we check that the both bacterial strain DS2 and RT12 also increase the chlorophyll B content under normal and stress condition. So after that, we also checked that for the proline content. So the proline content was find that it was increased under salt stress by both strain, and also the hydrogen peroxide content was reduced under salt stress. So these all were the characters that was bacteria shown, and the plants were to protect from the saline condition. So after that, we have concluded that RT12 and DS2 enhance cotton growth under normal and saline condition, and inoculation of these strains increase root, shoot length, and leaf development. Chlorophyll content was also improved, boosting photosynthesis, and it also showed that proline is also increased. H2O2 hydrogen peroxide decrease under salt stress. So the both strain showed strong halotolerance and plant growth traits. So it is a suitable thing as far as to use it as a biofertilizer for saline soil. Thank you, sir.
Thanks, Sajjad. Good job. Thanks for your presentation.
Yes.
You have got some questions, Professor Giver?
Yes.
Yes, I sent you, Sajjad.
Yes, sir.
You at first you can how to read the question.
First question, Sajjad.
换一个会议室，会议室时间到了啊。
Sajjad, this meeting time is up. We change another. Okay.
So again.
Okay.
Sorry. Okay. Sorry.
Okay. Sajjad, first question is how to isolate rhizobacteria. Can you explain the
Yes.
experimental procedure?
Okay.
Yes.
Okay. We have isolated the bacteria from the rhizosphere, and we have used the serial dilution method, and which we first take 1 gram of soil from the rhizosphere of the plant, and then we in a 10 ml tube, and 1 gram of soil we added 9 ml of distilled water. So after adding that water, we make 10 ml of solution, that is serial dilution solution, and we make 4 folds of that serial solution. So after that, we take it on the shaker. After shaking for 5 minutes, after that we drop a streaking of water in the tube, and of Luria agar 869 medium. After that, we drop streaking on another plates. So after that, they make colonies, and they have then we again and again use that method, and finally found a single colony. That was the method, sir.
The first is over.
Yes.
The second.
Second question, what is the basis for you to screen the strains with the salt concentration of 10 to 20% and treat a cotton plant 200 ml NaCl, sodium chloride?
Yes. We have used that to point out or to assess that does this bacteria show resistance to the salt tolerance or not. So after showing resistance to that bacteria and they were found salt tolerance, so for that purpose we assess that with that much salt, that is 200 millimolar. And we have already said in that presentation that they both strains were showed resistance and tolerance to the salt. So, but the DS2 were not shown that much on 20%. They were only showed on 10%. So that was the main base to point out that does this bacteria resist to that salt or not.
Okay, the third one.
Okay, third one. Were the salt tolerant PGBP strains isolated from cotton roots and or other environment? Investigate the impact on cotton metabolic.
Yes. We have been isolate the bacteria from the rhizosphere that was not endophytic bacteria, and we have just isolated it from the normal environment. But after that, we have used different methods to characterize this bacteria that does it plant growth promoting or not, and also that we have checked it that does it is a salt tolerance bacteria or not.
Then did you investigate the morphological and physiological indicators of cotton or conduct research on expression of salt tolerance gene?
Yes, we have just checked out the indicators of morphological and physiological, but we couldn't check the gene because our lab was less equipped. We don't have that equipment and our lab to check the gene expression. So we just checked out the morphological and physiological indicators.
Okay. The next question, explore to drought tolerance mechanism of PGBP in prosomillet. Which experimental approach would you prioritize in your study design?
Okay. If I found the opportunity to work on that research, so I will use different approach, but especially I will focus on molecular approach and physiological approach both. And so molecular approach, for that we should use the same method. We will identify, we will isolate the bacteria that is drought tolerant, and after that we should characterize it. And first, when we isolate, so if we want to check this bacteria that does it a drought tolerant or not, we will use in vitro screening of that bacteria. So for that we will use PEG, which is a natural occurring compound that is soluble in water, and they are specially used to lower the water balance. And at that condition, we will check that bacteria that does it drought tolerance or not. After that, we will characterize that bacteria that does it produce ammonia production, hydrogen cyanide, in different other things that is need for the plant growth. So after that, we will then inoculate the bacteria into prosomillet plant. So after that, after inoculated it into that plant, we will then using the molecular approach that does the bacteria change the molecular structure of that plants or not. So we will use proteomics of that plant and different genomic changes we will check out, and we will also check the physiological approach as well.
Okay, the last question you have. In salt tolerance activity experimental, which medium was used?
The medium was used Loria both LB agar medium. And we added 10% of salt and 20 and 15%. For different percentages, we added to that media. That is LB agar medium.
Finish. Oh,
Yes.
Professor, that's all. Any other questions?
No.
No. It's the time is up for the job. You can withdraw.
Yeah. Yes. So withdraw. And
Thank you.
Thank you. Thank you so much. Thank you.
Thank you.
Sanjay, you can leave. Yes.
Okay.